This is Bruce Jansen for IMNG Medical Media at the American College of Cardiology meeting. I'm with Dr. Matthew Price, who just presented the results of the GIFT exome study. Down the road, what do you see as the clinical implications?
Well, there's several clinical implications, both for this study and exome sequencing in general. I think in this particular study, a major question has been, we've found the CYP2C19 gene, but it only explains about 12% of the overall variability. You throw in clinical characteristics like diabetes and weight, you add a little bit more, but there's still a lot of unmeasured variability. I mean, or a lot of variability which is unaccounted for. So we need to do more work. This is preliminary, but genes such as the ones that we found likely explains more of that variability, and also can help us identify patients who may be at higher risk for events early after PCI. And that's something that we now need to go back and look at outcomes, early outcomes in patients who have had PCI, and confirm that variations in these genes are associated with outcomes. And once we get to that point, I think that changing therapy early on after PCI or during PCI, based on this genetic profile, may be something that we can consider.
Wouldn't it be possible to test the patient's propensity for non-response
exactly
off the drug before they've taken the drug?
Right, so that's the advantage of genotyping rather than platelet function testing. I've done a lot of work on platelet function testing. Platelet function testing is an important phenotypic test, but your patient needs to be on the drug to test your response. So if we can come up with a profile, a genetic profile, prior to exposing our patients to these drugs, I mean, that's really powerful. And now in the next year or two, we're going to have very rapid point-of-care genotyping platforms where from a single swab of saliva, you could get a genotypic profile within 45 minutes on very simple platforms, even just as simple as a USB chip to get somebody's genotype. So we're really moving forward very quickly technology-wise in terms of being able to adopt genotyping into clinical practice. And I think really what we need to do is really use these exploratory methods to look at new drugs, and even drugs that are on the market, look at adverse effects, like bleeding, for example, or other rare things that occur to our patients. And if we push for exploratory genomics using these high throughput techniques that have incredible power, we can really find the answers. And rather than just using very rote and rudimentary ways to determine the risk of our patients, diabetes or age, to really look at the genes and the proteins that cause these adverse effects and be able to adjust appropriately, I think would really be something important for our patients.